at the 8 o'clock hour. So, Magda, do you want to take it away? I know we're all so excited to hear this lecture.
Mhm. Oh, by the way, by the way, Melissa, I love your brain. So
Oh, thank
beautiful.
you. Yeah, it was by the help of ChatGPT.
Nice.
Oh.
Okay, so good morning, everyone. Welcome to once again a seminar series of the Unbowers Women's Brain Health Initiative. It is my pleasure to introduce today's speaker, Dr. Melissa Wright. Melissa is a postdoctoral researcher at Cardiff University Brain Research Imaging Centre, or CUBRIC, one of Europe's leading brain imaging centers. Over the past decade, she has developed an incredible research program at the intersection of endocrinology, vascular physiology, and neuroimaging. Her work asks a fundamental question: how do hormones shape the brain's blood supply throughout a woman's life? Using advanced MRI techniques and the lab's cutting-edge vascular neuroimaging toolbox, Melissa investigates how endocrine transitions influence vascular and neural health across both the brain and the eye. Her research spans the whole female reproductive lifespan, from the menstrual cycle to PMOS to menopause. Melissa is also a STEM for Britain 2024 finalist, has presented her work at the Every Woman Festival, and sits on the Society for Endocrinology Science Committee. Today, she will be sharing her work in this talk titled A Spotlight on Hormones and Cerebrovascular System. So please join me in giving her a warm welcome, Dr. Melissa Wright.
Thank you so much. And yeah, thank you so much for the invite to talk. It's such a great community. So hopefully I'll follow up your great other talks with something interesting. Yeah, my name's Melissa Wright. I'm an early career researcher. And so today I'd like to convince you that if we ignore the cerebrovascular system, we're missing an important part of women's brain health. And so first I'll just start with a quick introduction. So I'm from Cardiff University, which is here pictured a lot more sunnier than it usually is. It's usually a bit rainier than that. And specifically, I'm part of Kubrick, which is their brain imaging department. So the current building was opened 10 years ago by the late Queen, and as Magda said, it's one of Europe's top brain imaging centres. So we're really lucky in that we've got some amazing facilities here at Kubrick, including 5 different MRI scanners, which all kind of have their own specialty. We also have arranged electrophysiology scanning, such as EEG and MEG, and also cognitive testing, baby scanning facilities, etc. So yeah, we're really lucky here at Kubrick. And specifically, I'm part of Professor Kevin Murphy's lab. Here we are looking, here is a lot of the lab looking very cool in our matching t-shirts. And we kind of have two halves of the lab. So one half uses physics principles to develop new methodology for measuring physiology in the brain and beyond. And in the other half of the lab, we apply those methods to investigate vascular physiology across aging, neurodegenerative conditions, and endocrine states. So why should we care about the cerebrovascular system? So as you'll probably know, the brain is an incredibly energy-hungry organ, and it consumes 20% of the body's oxygen despite only being 2% of the body's mass. And in order to function normally, sort of everyday cognitive functions, the neurons require a constant, steady blood supply in order to provide them with resources and take away waste. But beyond that resting blood flow, vessels also need to react dynamically, and this ability is a key marker of cerebrovascular health. So following neural activity and neurovascular coupling processes, blood vessels dilate, leading to an increase in local blood flow to meet that increased energy demand. And this is one of the basic foundations for fMRI. And another important aspect is vessel stiffness. So vessel walls need to have a baseline level of elasticity in order to absorb pulsatile energy coming from the heart. If the vessels are stiff, which can happen with old age, this energy isn't fully absorbed by the time we get into the small cerebral capillaries, and that causes damage, which can be seen as white matter hyperintensities. And they're a potential mechanism for cognitive decline as it's very related to the aging process. So you can think of the vascular system as the brain's plumbing. Even if the house is fine, problems with the pipes can affect everything. And damage or alterations to the healthy vascular system are associated with a range of different conditions. And this is quite nicely shown with dementia, where changes in cerebral blood flow is one of the earliest alterations, so the earliest markers of the disease. And just quickly, I'll go over how we measure these different things with what we call our cerebrovascular toolbox at Kubrick. So MRI could be used to non-invasively image vascular structure, function, and physiology, from the microvascular to the macrovascular. So these vessels throughout the cortical vascular tree have a range of receptors that impact vasodilation, including hormone receptors. And so I'll start with the microvasculature, which are the smaller vessels that permeate the cortex. And the most common way of analyzing this is using ASL. And this is kind of the bread and butter of physiology imaging, where we use both a labeling and an imaging plane to map perfusion across the cortex. Depending on the type of ASL, we can measure cerebral blood flow, which is the amount of blood flow into the cortex. And we can also measure arterial arrival time, which is the speed of blood flow in the cortex, which can be affected, for example, by arterial stiffness. And this is found to be really sensitive to various conditions and also the aging process. So there's also cerebrovascular reactivity, which is that dynamic aspect. So it's the dynamic ability for blood vessels to respond to a vasoactive stimulus. And like I said before, really supports neural activity. So we can measure this either using CO2 gas delivery, which is kind of the gold standard. But we can also naturally rise the blood CO2 level using breath hold methodology, which tends to be a bit more tolerable to sort of patient communities or older age. We can also look at the cerebral metabolic rate of oxygen. So this is the rate that oxygen actually metabolizes in the tissue. And kind of the gold standard for measuring this is again a gas delivery. And using that, we can map oxygen delivery throughout the brain at a really high spatial resolution. But you can also do this quickly using what's called a truss scan, which is just a couple of minutes, you get a single value for the whole brain. So that's kind of a nice and easy one that we tend to put into studies. And again, it's a bit more tolerable than having the gas challenges. Another aspect is cerebral autoregulation, and this is another one which tends to fail with the aging process. And this is basically the ability for the cerebral vasculature to adjust to different levels of blood pressure. So it's kind of maintain that consistent, steady blood flow despite changes in blood pressure. And we can look at this adaptive ability by using blood pressure challenges. So kind of the scary coffin-like thing in the corner is what's called a lower body pressure technique. And that increases blood pressure, which then we can see the sort of adaptation in the vessels. And finally, another one that we use within the lab is blood-brain barrier permeability. And this is especially relevant for when you're thinking about sort of symptomology. It's been associated with levels of brain fog in, for example, COVID. So it's definitely going to be really interesting to apply that to certain endocrine states. So those are all kind of the microvasculature, but some of the people in the lab have been developing ways to look at the macrovasculature, which would specifically allow us to look at certain vessels and see how they function. And in particular, that's looking at the pulsatility of the vessels, that kind of pulsatile energy that I mentioned before. So Ian Driver in particular has been developing some really cool methods for measuring pulsatility called Dimax scanning. This images the vessels at an incredibly fine temporal resolution and is able to capture that beat-to-beat pulsatility. This is my favorite MRI scan to acquire because it's really cool seeing that real-time pulsatility. When we then compare the pulsatility timing between vessels, we can then estimate the speed of the pulse throughout the cortical tree and thus infer vessel stiffness. And so far we found that this is sensitive to age, so with stiffer vessels with increasing age. So why should we care about any of this in the context of women's brain health? We know from animal models that there are ovarian hormone receptors throughout the brain, including cerebral vessels, which is what these images are showing here. And this figure from a few years ago really nicely shows the wide variety of physiology that have been associated with sex hormones in animal models through mechanisms such as increased nitric oxide bioavailability or changes to the endothelial function. And through these factors, you can see how hormones have the potential to have a really dramatic effect on brain health. But how does this relate to hormones across human endocrine states? So obviously, I don't need to explain to this audience that ovarian hormones fluctuate hugely across an average woman's reproductive lifespan. We have the increase of ovarian hormones and kind of the start of menstrual cycle with puberty and menarche. We have the regular fluctuations in hormones with the menstrual cycle. If pregnancy occurs, that's a huge 1,000-fold increase in these hormones. Then finally, with menopause, you have the dysregulation and eventual decline. And the impact of these hormones on brain health is really demonstrated by how closely tied disease risks are to these endocrine transitions in terms of mental health, neurodegenerative conditions, and vascular disease. Such risks have also been associated with alterations in these endocrine rhythms. So here's a few examples. So for example, a history of irregular menstrual cycles has been linked to cerebrovascular disease outcomes independent of any of the risk factors. Similarly, PMOS, which is characterized by an altered endocrine profile, is also associated with a greater lifetime vascular disease risk. But the exact details of these mechanisms in humans is still relatively underexplored. So that's something that we've been looking at in our lab. So we began by investigating the menstrual cycle. This is a great model for this because we have these short-term, relatively predictable changes in hormones that we can investigate in a otherwise healthy cohort. So the aim of this first study was to investigate the influence of hormonal fluctuations across the healthy menstrual cycle using both cortical and retinal resting vascular functions. So we had a cohort of healthy menstruating women, which we sampled 3 times across the cycle. So this wasn't to compare menstrual phase per se, but it was to sample varying levels of hormones in each participant. So we looked at the early follicular phase, the late follicular phase, and the mid-luteal phase. And we used different methods to look at both the brain and the eye. So for the brain, we used 3T MRI. We looked at perfusion, so that's the cerebral blood flow and arterial arrival time. We also looked at choroidal artery pulsatility and also some oxygen metabolism measures. And we also use a technique called OCT angiography to look at the eye. And these are really, really complementary measures because we can see the vasculature at such different scales. And from these retinal images, we can get the vessel density, and we can also get the retinal blood flow resistance. So to explain that a little bit more, to analyze the retinal images, we use in-house software from our collaborators at the University of Edinburgh. So this is the raw image that we get. And you can see just by looking, we get this really fine detail. So these scans are 3 millimeters by 3 millimeters. So we can see these really tiny vessels that we wouldn't be able to do so with the MRI. And we then binarize the images. And from this, we can get the vessel density just by looking at the ratio of white to black pixels. We then create a graphical representation wherein each node contains information about the radius and the length of the vessel. And from this, we can estimate mean resistance of the blood from each of these different ROIs. So we analysed our cortical and retinal outcomes using linear mixed models. We bought both estrogen and progesterone, which were measured using blood samples in the study. So then we could account for the different effects that these have. And so what we found was that oxygen metabolism and cortical pulsatility seemed to be fairly stable across the menstrual cycle, but we found these changes in cortical perfusion. So specifically, both estrogen and progesterone were associated with an increase in resting blood flow and a decrease in arterial arrival time. And so these values are the average across a whole cycle. In terms of the retina, we found that the progesterone wasn't significant, and vessel density also seemed to be quite stable. But estrogen was associated with a decrease in blood flow resistance. So this would be an increase in blood flow. So it suggests that estrogen in particular was associated with increase in blood flow across both the brain and the eye. So these different vascular beds and these different sizes of vessels. But as I said before, another aspect of this is the dynamic vascular function, which is also really important for everyday functioning. So in the same cohort, we ran this second study where we aimed to investigate this dynamic cerebrovascular function using MRI. So firstly, we looked at cerebrovascular reactivity, which is the ability of blood vessels to dilate in response to a vasoactive stimulus. And in this case, we used CO2. So we measured perfusion during normal air inhalation and also during 5% CO2 delivery. And we defined this both as the increase in blood flow and increase in the Bold signal. We also looked at the hemodynamic response function, or the HRF, which is another useful metric for interrogating the dynamic functionality of the vascular system. So using Bold fMRI, the Bold signal is mapped in response to a stimulus designed to evoke a strong but brief neural response. So the final HRF shape contains information about both the neural activation, the cerebrovascular coupling mechanisms, and the local blood flow increase. And while it's not possible to detangle these different contributions, it gives you a nice overview of the whole process. So again, this was the same cohort as before. It was a slightly smaller sample because we used gas delivery, not everyone wanted to do that. We measured estradiol and progesterone using bloods, and all participants fasted before testing. And we used 3T MRI, and we analyzed the results using the linear mixed models. And what we found that for both estradiol and progesterone were associated with a small but notable increase in cerebrovascular reactivity, which has interesting implications both for menstrual symptoms, but also for other endocrine conditions when you might have greater changes in hormones. And in terms of the HRF shape, we found that this also had a change with the different hormonal conditions. So estradiol was associated with slightly larger and earlier HRFs, while progesterone was associated with slightly wider HRFs. And again, while it's not really possible just from this to detangle what the mechanism was behind that, we can make some inferences. So for example, the slightly increased peak amplitude that we get from estradiol might be related to the increase in cerebrovascular reactivity that we saw before. And so these results have implications for how hormones impact how well the vascular system supports neural activity throughout the reproductive lifespan, but also has implications for fMRI. And as I'm sure a lot of you know, fMRI uses a complex signal based on neurovascular coupling to make inferences about neural activity. And a lot of standard fMRI analysis pipelines involve HRF convolution of the simulated or event-related time course. So if there are any systematic differences in HRF shape between groups, potentially because of an endocrine difference, then that might bias the results or be introducing noise. So as a secondary experiment, we therefore aim to investigate this by investigating the impact of either using a standard canonical HRF during an analysis pipeline or using an individually measured HRF. And we did this on a task called population receptive field size mapping. So I'll just really quickly introduce what that is. So every neuron in our visual cortex responds to a certain amount of the visual field. That's its receptive field size. But these are much too small to measure individually. So what we have to do is take all of the neighboring neurons, which will respond to neighboring parts of the visual field, and measure all of them within a voxel. And that overall area that they respond to is the overall population receptive field size. So this has been related to visual attention and various visual symptoms. And it's also theorized to be related to sort of dendritic tree size and that kind of thing. So yeah, we plotted these across the cycles, but then we put them into our hormonal models. And we took this from visual area V1 and visual area V2. So what we found was when using the canonical HRF, it seemed to be quite stable across the menstrual cycle. And that's what we found when we put it into our hormonal models. But when we used that individually mapped HRF, we saw that it seemed to be this change across the menstrual cycle. And when we put this into our hormonal models, we found that estradiol was associated with an increase in PRF size, whereas progesterone was associated with a decrease, which is especially interesting considering some findings of visual acuity changes across the menstrual cycle, and also changes in dendritic tree in other parts of the cortex with estrogen. So what we found from these two studies was both that ovarian hormones influence both resting and dynamic vascular function in the brain. And this must be considered when using methodologies such as fMRI, which assume equal vascular contribution to make inferences about underlying neural activity. But there was a limitation to this. So hormones vary day by day. And if we just use this coarse measuring, so 3 times across the cycle, we miss a lot of variation. And I'm going to be preaching to the choir here because I'm sure a lot of you know about this, but the way a lot of people have been looking at this is using high density sampling, where you sample just a small number or just one person multiple times across the menstrual cycle. And you can really look at that find day-to-day fluctuations in hormones and what's going on in the brain and sort of couple them together. So it's a really powerful technique for mapping individual neuroendocrine sensitivity profiles. And we know from other amazing work, such as by Karina Heller, that the exact profile can vary with cycle regularity and mediate mood symptoms. So while it's just in a single person, this kind of work is a step towards understanding the neural underpinnings of specific hormone sensitivity profiles, as well as generating specific hypotheses. So we ran what was called the 100 Days Project. It's basically a high-density project using all of our cerebrovascular measures. As you can see, I was the participant, where I scanned myself every other day, almost for 100 days. And during each testing session, we started with a morning saliva sample. So I'm very happy by the time I get to the end of it. We started with a saliva sample to look at day-to-day hormones. We then did the MRI scan. We then did a physiology scan, which was kind of blood pressure and systematic vascular function. We did an eye scan, so that was the OCT that we looked at before. And then finally, for every single day, we did some questionnaires. And we did menstrual symptoms, mood questionnaires, and sensory sensitivity. So this just gives you a quick overview of that data set. We see from this figure at the bottom, I don't know if my mouse is showing up, but we covered 3 full menstrual cycles and kind of clipped one before and after. We had 43 test sessions where we had hormones and the MRI. Unfortunately, the OCT decided to break when we started the study, so we've only got half the amount of OCT images, unfortunately. So this is actually the first time I'm presenting these results, so it's hot off the press. And we're still in the process of finalising all the models. So if you have any thoughts or ideas, I'd love to hear or love to have a chat about it. And during this talk, I'll just focus on these first 2, but watch this space for the rest. So firstly, looking at the participant characteristics. So in these were saliva samples of the hormones, and we've collapsed this across menstrual cycle day, and you can see we sort of saw what we expected, which is good. We had those stool peaks of estradiol and the later peaks of progesterone. Testosterone was quite stable across the cycle, but sort of had a gradual increase. In terms of the symptomology, so the DRSP is our menstrual symptoms. So that includes both physical and psychosocial symptoms. Then we have negative affect and positive affect. And you can see from the figures that there is some hormonal sensitivity. So we have greater sensitivity, by which I mean more menstrual symptoms, a more negative affect, and less positive affect during menstruation and also during ovulation. And I think this is important to kind of put the results in context when you remember it's a single sensitivity profile that you're mapping, and how this varies might vary from symptom profile to symptom profile. So we found that we kind of replicated some of our results from the other study, which was exciting. So we found that estrogen was associated with an increase in perfusion, so that increase in cerebral blood flow, and also an increase in cerebrovascular reactivity. Progesterone, we had that increase in cerebral blood flow and in cerebrovascular reactivity. But we found that for arterial arrival time, we had a small increase with estrogen, but no significant effect with progesterone. So that was a bit different than from our group effects. And testosterone, we found it had a negative effect through all our perfusion measures and the cerebrovascular reactivity. And to say we put these in a model all together so we could kind of account for the effect of the other hormones. In terms of our oxygen metabolism measures, that seemed to be pretty stable across the menstrual cycle, again, replicating what we had in our group study. And the blood-brain barrier and cortical pulsatility are currently being analysed, so watch this space. And what we're planning to do next with this study is we're going to look specifically at the hippocampus, because it's really dense with these ovarian hormone receptors, and also look at how changes in these measures are relating to the vascular function across the body and the symptomologies that we've got. Then if you want to talk about this, then yeah, just let me know. And finally, so we've got a couple of studies that are currently running that I'll quickly introduce you to. So what I really wanted to do following this was link some of these MRI measures that we've taken to symptoms that participants are actually experiencing and different kind of sensitivity profiles. So we've been doing this with two different methods. So one that we've called NESI, which is the neuroendocrine symptom sensitivity imaging study. And this is with participants who report severe menstrual symptoms. We're following them across 2 cycles and looking at how vascular and microstructural changes relate onto the symptoms they report. We're also doing the MindFog study, which is looking across menopause, and we'll be investigating how these changes in cerebrovascular function, especially things like blood-brain barrier, relate to their reports of subjective cognitive decline, such as brain fog. So yeah, these are currently collecting, so it's very exciting. And finally, I'll just leave you with a little take-home message. So women's brain health is often considered through a neural lens, but by adding in these vascular factors, we might gain a more complete understanding and uncover mechanisms that explain both regular endocrine states and vulnerability to symptomology and disease across the lifespan. And lastly, I'd just like to say a quick thank you to the amazing lab at Cardiff, all of our collaborators. And yeah, thank you very much for this community and for inviting me.
Thank you, Melissa. Um, any questions for Melissa?
Yeah, Melissa, that was fantastic. If just to echo Magda, if anybody has questions about her work, you can throw it in the discussion or in the chat. I'll kick off with one. So it's such a pleasure to see how this work is developed and to have a front row seat to so many of these incredible ideas. You've done just a brilliant job, and there's that 100-day study. You have so much more coming down the pike, I know, so I can't wait. And I'm sure I speak on behalf of everybody here. We can't wait to see what you find. Question for the field, given what you found with sort of the stretching, the sort of modulation of the HRF in response to these gonadal hormones, what would be your advice to neuroimagers who maybe aren't neuroendocrinologists, aren't thinking about hormones? Do we need to do individualized HRF? Do we need to think about, hormone regulation of this in our models, even if you're not, again, focused on kind of hormones per se? And then second to that, can you tell us about, because I know you saw some testosterone effects there, and I'm wondering if we can either use an existing data set or just generate a new one to test maybe those diurnal changes in testosterone and its effect on HRF. Because The motivation there is to not leave people with this sense that like women are noisy and shouldn't be MRI participants because their HRF is changing, but probably there's like all these like baked in factors that we've just ignored, and it's true for all sexes and genders. But yeah, tell us what we should do as good methodologists.
Yeah, so I think the gold standard would be to sort of measure the HRF in the study, I think. And especially if you're just doing a visual HRF, it's quite quick to do. I think ours was 4 minutes, I want to say. But yeah, I think it's just one of those things that this is a additional thing that's going to contribute to noise. So if if you can just ask about the what's the average cycle length and cycle day, then that'll be a bit of information you can add in to try and compensate for that variation. So it's like a lot of things like caffeine intake can vary it as well. But it's just one of the things that you can add that sort of that will help with these kind of thing. And especially if you're looking at group studies where you might expect a hormone difference, I think it's a bit more important to try and account for those differences.
Mhm.
So in terms of the testosterone, so yeah, that would be really interesting. And I think, yeah, it definitely has the potential. I mean, everyone has all of these hormones. I'm sure they have an equally dramatic effect in males as well. Just we have these nice endocrine states with women that we could study.
Mhm.
And yeah, I definitely don't think it's just a women's problem. So it would definitely be interesting to sort of map this across like I said, across the diurnal cycle to see what effects that they have.
Yeah.
So I have a couple questions. First of all, it was really super interesting. I think there's lots of implications for our fMRI studies that we always do, right? So, but I have a couple questions. Yeah, I think you mentioned that you'd like looked at region level metrics. Were the things that you were showing mostly like average global values or?
Yeah, so sorry, I should have mentioned that we looked at across different atlas areas, and what we found was that the average effect across kind of globally didn't differ significantly from any particular region.
Okay.
So it seemed to be a global effect.
Global effect, yeah, which would make
Yeah.
sense, yeah. And then the other thing I had, a question I had was that there's this link between cerebrovascular reactivity and like lymphatic clearance. Do you think that might be a
Yeah.
potential mechanistic link between hormone level changes to the vascular system and like accumulation of misfolded proteins or?
Yeah, I think that's a really interesting question, and it would definitely be interesting to look at. So yeah, they're really sort of tied to each other. And so Hannah Chandler in the lab is actually using some of that pulsatility to start
Mhm.
looking at CSF flow,
Mhm.
and sort of from that can infer something about lymphatic systems.
Mhm.
So yeah, down the line, it'd be really interesting to see how that varies with endocrine state. And it
Yeah,
would definitely make sense that there would be some kind of relationship there.
very, very cool. Thank you.
And I wanted to ask what drew your attention to the eye, to studying the eye as well as the brain. Is
Yeah,
it?
well, I actually did my PhD was more eye related in vision neuroscience. And then kind of changed field a little bit and started doing the neuroendocrine stuff during my postdoc. Yeah, I think it's really nice because it's a very complementary mechanism because we can see such fine detail that we can't see with the MRI. And it
Mm,
also has nice sort of clinical implications because it is one of the only parts of the central nervous system that you can see with the naked eye. So if there were some systematic differences with OCT and with the retina, then that could be really easily checked in like a optometrist visit, for example.
interesting. Yeah.
There's another question from the audience when it comes to sort of what drew your attention to different areas. Somebody asks why the hippocampus rather than, say, the hypothalamus.
Yeah, the hippocampus we chose for looking at this just because it is so dense with sex hormone receptors and it's also been applied a lot in the literature. And I also think with tying it into some of our mood symptoms and psychosocial symptoms that we're measuring, it will be interesting to make that link. But actually, so Rebecca Oates, who I think I'm working with on the Nessie study, So she's interested in sensory sensitivity, and we're hoping to look at the hypothalamic changes and how that might relate to these sensory sensitivity changes across a cycle. So that's kind of another branch that we've been looking at with the high density study.
Very cool. There's another question about how subjective measures are going to be measured in the mind fog study, and then a question about sort of thinking about race and ethnicity and how to generalize these data given differences in menopause symptoms that can map on across different demographics.
Very good question. Now, in terms of how to measure subjective brain fog, that is something that's quite tricky. So there are a few different questionnaires and sort of ways of looking at it. And there's the kind of argument of, do you have brain fog in different conditions? Is that all sort of describing the same phenomenon, or is it different per each one? So we chose two different questionnaires that was focusing on different aspects of brain fog. So we'll be specifically kind of looking at reports of those kind of symptoms. So one of which is the brain fog scale, and another one is the everyday memory questionnaire. And from those, we can look at different domains, such as mental fatigue and also sort of everyday memory. And in terms of the race and ethnicity, that's a really good question. And that's definitely one of the issues, I think, with some of our samples, is they're quite homogenous. But I would really like to sort of introduce that a bit more, because I think it would definitely vary a lot, especially when you have cerebrovascular risk, for example, varies by race. So that's definitely something I would be really interested to look at in the future. And it's something that we're sort of measuring in the brain fog study, but yeah, we'll see how the sample goes.
You're just getting started. There's one clarification question from Nina Kozlov. She asks, so testosterone levels were inversely associated with measures of blood flow. Can you tell us maybe a little bit more about those testosterone findings?
Yeah, so they were associated with a decrease in perfusion, so the cerebral blood flow, also a decrease in the arterial arrival time and in cerebrovascular reactivity. So it kind of had the opposite effects in estrogen in all of these factors, but then it didn't influence oxygen metabolism either. So it would be interesting to see if this is the case in other kind of symptom profiles. For example, in PMOS maybe, where we have these a different endocrine profile where they have a lifetime exposure of greater testosterone levels. Do we see the same effects?
And following up on that, Isabella asks, given the effects that you find with testosterone in the cerebrovascular system, how do you think this will influence testosterone therapy for postmenopausal women? So there's this sort of, hype around testosterone pellets, which I think most people would say is really not clinically guided because you can't take them out. They live in you for months on end. But this is something that, is being talked about sort of by menopause influencers, and there's other forms of testosterone treatment that maybe are a little bit more clinically guided, but it's certainly an unknown, and I don't think anybody in this space is thinking about those implications of menopausal hormone therapy on the cerebrovascular system. So given your work moving into menopause, tell us, what the implications of this are.
Yeah, I think that's really interesting. Yeah, it'd be really interesting to have a look at that, because this is definitely kind of a few stages down the line. These are quite small changes in testosterone just across the menstrual cycle. So it'd be really interesting to see sort of how that changes when we're having these larger effects, and when it's in the context of sort of menopause and the kind of dysregulation of hormones. For example, and we also have people who have gender affirming therapy, who having testosterone through that, so having a younger kind of system. It's another area where there hasn't been much work into it, but you definitely have some implications. For example, people who have that have a lower rate of migraines, which might be vascular related. So there's a few kind of interesting hints that the vascular system is being affected, but yeah, it's really not been hugely looked at.
Well, any final questions for our incredible guest? Okay, it looks like we have one. A great talk, Melissa. In your opinion, what is the evolutionary advantage of women having changes in cerebrovascular structures during menstruation?
That's an interesting question. Yeah, it's an interesting question. I've never really thought about the evolutionary perspective, but yeah, I'll have to have a think about it. You do have, yeah, you do have kind of knock-on effects on things like visual acuity. You might imagine sort of some of the performance changes you might get, but yeah, to be honest, I haven't really thought about it before. It's only something to think about.
A good one for the group to chew on.
Yeah.
Um, well, thank you so much for giving us your time. And I will just make a plea to this audience that I think the work that you, Melissa, and the group at Kubrick are doing is just pushing this field into new territories. And I know you're incredibly collaborative. So, if folks have any questions about the cerebrovascular system, best practices and how to measure it, how to think about pulling this into the women's brain health space, which is just so ripe for discovery. I'd say you are such a perfect point person for the field. So reach out to Dr. Wright, and I think we're all looking forward to seeing the incredible work that's going to be coming out through future publications. So thank you for moving us into this space. It's a long time coming, and you're just the perfect person to be leading this charge. So thanks for sharing your wisdom with all of us.
Oh, thank you so much. And yeah, thank you for inviting me. It's yeah, such a great community. And yeah, please do
Awesome.
reach out. I'd love to keep in touch with anyone.
And probably we showed this in the kind of early slide deck, but we're taking a break for August. So we'll see everybody back here at the watering hole in September. We have an incredible lineup of speakers coming up for next term. So have a great summer. Stay cool, everybody, and we'll see you in the fall.
Bye. Thank you. Thank you so
All
much.
right.
Bye
Bye.
bye.
Ciao. Bye.